Good afternoon, everyone. Welcome to the SDB Ethel Browne Harvey Postdoctoral Seminar Series. My name is Ryong Kwon, a postdoc at Oklahoma Medical Research Foundation, and we'll be moderating today with Dr. Leslie Slota-Burtt, a postdoc at Duke University. We are very excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Lydia Grmai at University of Pittsburgh and Dr. Donn Van Deren at University of Utah School of Medicine will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. It is my pleasure to introduce our first speaker, Dr. Lydia Grmai. Dr. Grmai received her PhD from New York University, studying sex determination pathway using Drosophila as a genetic tool in Dr. Erica Bach's lab. And she joined to Dr. Mark Van Doren's lab as a postdoc at Johns Hopkins University to continue to her study sexual identity-related questions using Drosophila model. She's now a postdoctoral associate in Dr. Deepika Vasudevan's lab at University of Pittsburgh, and also co-mentored by Dr. Kapu Durasa at Duke University. Dr. Grmai is currently focusing on investigating the role of cellular stress and inner organ signaling in reproduction, as well as developing genetic tools to explore the basis of heterogeneity in ER stress response. She received the T32 award and was recognized as leading edge fellow from Howard Hughes Medical Institute. Today, she will be presenting her current work on the stress response factor ATF4 in adipose tissue acts as a systemic regulator of reproduction. Without further ado, I present to Dr. Grmai.
And can everyone hear me and see okay? Yes. All right, well, thank you so much for the introduction. As he said, my name is Lydia Grmai. I'm a postdoc currently working jointly between the University of Pittsburgh as well as Duke University. And I'm grateful to the SDB for giving me the opportunity to share my work with you today on the role of the stress response factor ATF4 in adipose tissue in regulating reproduction. So I've been interested for a number of years in understanding how reproduction is regulated. And a lot of that work over the years has been focused on gonad autonomous signals and gene regulation events that ensure the healthy production of gametes. But more recently, what I've been focusing my research questions on is this idea that reproduction is actually not something restricted to gonad signaling, but that it's actually a systemically coordinated process that requires inputs from multiple tissues, including the brain, adipose tissue, intestine, and certainly others as well. And today I want to focus on the relationship between adipose tissue and the ovary. And as we know, adipose tissue is very lipid rich, and clinically differences or changes in lipid content have actually been linked to changes in reproductive output. And this is both in the case of low lipid content as well as high lipid content. Both have been shown to cause defects in ovulation, defects in fertility. And so some of my questions recently have been focused on understanding how an organism can sense these differences or changes in lipid content, and then how that information can then be relayed to the reproductive organs to alter the rate of reproductive output. And a lot of this work has been done in the last year or so, year and a half, in the lab of Deepika Vasudevan, who's at the University of Pittsburgh. And our lab's central focus is understanding how this pathway called the integrated stress response regulates or is implicated in both homeostatic and disease contexts. And so this pathway can be activated from a number of both internal or external stress cues. And this will lead to activation of one of the integrated stress response kinases in Drosophila, either PERK or GCN2. And activation of either of these kinases will culminate in the induction of this transcription factor, ATF4. And then ATF4 can then go on to activate transcriptional programs to help alleviate the stress cue or stress event. And so we actually know that in adipose tissue, there are basal levels of integrated stress response signaling, and as evidenced by the induction of ATF4, likely because of the high metabolic burden of these cells. And so the work I want to describe to you today is really looking at the role of the integrated stress response in fat tissue in regulating reproduction. And so this project really got started when we first noticed this fertility defect in animals that were globally mutant for ATF4. So using alleles that are hypomorphic for ATF4 activity or expression, we noticed that in an egg-laying assay, animals that were homozygous for a hypomorphic allele of ATF4 showed reduced egg laying in a fertility assay. And so this is just quantifying the number of eggs that are laid per female per day. And you can see that compared with controls, ATF4 mutants lay significantly fewer eggs. And additionally, many of these eggs exhibited morphological defects that we know to be to correlate with a decreased likelihood that these eggs will hatch into healthy larvae. So to try and understand where this fertility defect might be happening, we looked further into their ovaries to see if we could find any more specific defects in the process of oogenesis. So just to briefly orient you to the process of oogenesis, this schematic depicts one of the two ovaries that would be in an adult female. And each of these ovaries consists of a number of these ovarial chains. And so the chains consist of maturing egg chambers or follicles at one of 14 maturing stages. And so each of these maturing follicles will contain exactly one future oocyte. And that's shown by this dark gray cell, as well as a number of supporting germ cells shown in light gray, and a number of supporting somatic cells that are located in the gonad as well. And so this process of oogenesis will culminate with stage 14, which is the mature oocyte. And you can see what that looks like in a confocal microscopy image, where you can see follicles maturing at different stages, and DAPI marking all of the nuclei. So one of the first things that we noticed in these ATF4 mutant ovaries was that instead of being able to see kind of egg chambers at multiple stages of development, we started to notice that frequently ovarial chains would arrest at a stage around mid-oogenesis, where it wasn't always clear that there was an oocyte located at the end of that ovarial chain. And so we were able to quantify this, and we see that ATF4 mutant ovaries more frequently exhibit this mid-oogenesis arrest. So at this point, we wanted to try and understand what tissue ATF4 is required in for oogenesis. And so using a GFP knock-in reporter for ATF4, we looked at the ovaries from these animals, and we actually saw that we couldn't find ATF4 protein in the ovary proper. And so we looked to see if maybe ATF4 expressed in another tissue might be relevant in this context. And we saw that in neighboring adult adipose tissue that we could see using the same GFP reporter that now the nuclei of fat tissue or adipose tissue clearly shows expression of ATF4. And which is consistent with what we know about ISR signaling, integrated stress response signaling, that there is basal ATF4 activity in healthy fat tissue. So if we think about anatomically where the fat tissue is located in the animal, Sheets of adipose tissue in the adult fly line the inside of the cuticle of the animal. And since the ovaries kind of occupy a lot of the abdominal space in an adult female, the ovary is sort of surrounded from all angles by fat tissue. And so this makes it kind of a likely candidate for non-autonomous signaling to the ovary. So to see whether ATF4 specifically in the fat is required to support oogenesis, we utilized the Gal4 UAS binary expression system, which is used very widely in Drosophila research, as I'm sure many of you know, by utilizing a fat-specific Gal4 line. This is a promoter or an enhancer element that is known to be fat-specific. And then crossing those animals to those expressing or containing an ATF4 RNAi construct downstream of Gal4's binding site or UAS. So now in animals that inherit both Gal4 and UAS, we essentially will get fat body specific depletion of ATF4 protein. And so in these animals, we noticed that we were able to reproduce the fertility defects that we saw in the global ATF4 mutants, where we see a decreased number of eggs laid, as well as an increase in the number of those eggs that show these morphological defects. And so this really tells us that fat body specific ATF4 is required non-autonomously to support oogenesis. Another thing that we noticed in these mutant ovaries is that if you just look at the morphology of the nuclei by DAPI staining, we can see that in global ATF4 mutants, frequently we were able to observe follicles or egg chambers that seem to be undergoing cell death. And so you can see the sort of structural integrity of these nuclei have kind of disintegrated. And we noticed the same examples of follicle death when we just depleted ATF4 from the fat. And I've outlined a few of these follicles here. And interestingly, we saw that this follicle death also happened if we simply depleted either of the kinases that activate ATF4, so PERK or GCN2. The knockdown of either of these in the fat body also led to this follicle death. And so this really suggests that the integrated stress response is acting in fat tissue to regulate oogenesis or to promote oogenesis. So in trying to understand what specifically the role is of ATF4 in the fat in this context, we kind of thought more carefully about when we're sort of seeing these phenotypes emerge in the ovary. And the follicle arrest or follicle death always seemed to happen around mid-oogenesis. And that timing, developmental timing in oogenesis coincides with the onset of a process called vitellogenesis. And this is a process where yolk proteins and lipid particles are synthesized in somatic cells and then trafficked into maturing oocytes. And these particles are going to be required in the mature oocyte and beyond to support early embryonic development. And so these yolk proteins are synthesized both by somatic cells within the gonad that are surrounding the oocyte, as well as from the fat tissue. These are both major sources of yolk protein. And we know that vitellogenesis or yolk protein trafficking to the oocyte is supported or is mediated through lipoprotein particles. So these are yolk proteins, lipoproteins that are coating lipid droplets. And so these lipid droplets form in the somatic cells, are coated by yolk proteins and then are trafficked to the ovary. So to see whether ATF4 is regulating vitellogenesis or promoting vitellogenesis, we first just looked at the mature oocytes from these animals when we deplete from control animals or when we deplete ATF4 from the fat. And so here you can see mature oocyte from a control animal, and these dark spots in the oocyte represent the yolk granules that have been uptaken by the germline. And you can see that in oocytes from animals where we depleted ATF4 in the fat, you can see that there are fewer granules, and many of those granules appear smaller, suggesting that yolk protein trafficking into the oocytes is impaired when we get rid of ATF4 in fat. And we can actually see what these yolk proteins look like in fat tissue using a yolk protein GFP fusion construct, where in control fat tissue, you can see an abundance of yolk protein punctae. However, when we deplete ATF4 from the fat, we now see that there is a dramatic reduction in the abundance of yolk proteins within the fat tissue. And since I mentioned that these yolk proteins are trafficked to the ovary as lipoprotein particles, we wanted to see if lipid droplet formation might also be compromised in these fat tissues. So here, just using a dye that stains neutral lipids called Bodapy, we can see lipid droplets of varying sizes in control tissue, fat tissue. And then when we deplete ATF4 in fat tissue, what we noticed was that we see lipid staining, but the cells don't seem to be able to form these lipid particles as efficiently. And so it seems that ATF4 possibly also regulates the formation of lipid droplets in the fat body. So what I've shown you so far is that ATF4 is required specifically in fat tissue to support oogenesis, and it does that in part by promoting vitellogenesis through the production of yolk lipoprotein particles to then traffic them to the ovary. So I want to spend the rest of my time talking about a second interesting phenotype that we found in these same animals. So if we look at control ovaries, these are just two ovaries isolated from one control animal. You can see at the base of each ovary the presence of these mature oocytes in each ovary. And what we noticed is that in animals where we depleted either PERK or GCN2 from the fat tissue, their ovaries seem to contain many more mature oocytes on average compared with control. And this initially seemed kind of counterintuitive, since I just explained to you that loss of either of these factors actually reduces the rate of oogenesis. And so it seemed paradoxical that we are now seeing more eggs per ovary. And so we wondered if perhaps the rate of oogenesis is reduced, but the animal still can make some eggs, but now perhaps they are less efficient at laying those eggs. And so we wanted to assay whether ATF4 is actually also acting in the fat to promote reproductive behavior in the way of ovulation or egg laying. And so we tested this first just by blocking secretion from the fat by overexpressing a particle of tetanus toxin protein, which is the toxic agent of tetanus that will block the secretory pathway. And we see what we call egg retention, or the presence of more mature eggs per ovary compared to a control ovary, just when we block secretion from the fat. And so we wanted to see if maybe there is a specific, neuropeptide or other protein that we could identify downstream of ATF4 that regulates ovulation. And so we were able to take advantage of a publicly available ChIP-seq data set generated by Kevin White at U of Chicago, where he probed ATF4 occupancy in the Drosophila embryo. And so we found a number of neuropeptides that seem to have ATF4 occupancy somewhere within their locus. And we were actually able to identify the peptide CNMamide or CNMA as an ATF4 regulated neuropeptide in the fat body. So using a reporter for CNMA, you can see that it is expressed in the fat tissue. These are two Drosophila larvae where we can see CNMA in the fat. However, when we knock down ATF4 in this background, we see a reduction of this reporter, suggesting that CNMA is actually downstream of ATF4 in the fat. And then if we just deplete CNMA in the fat on its own without disrupting any other genes, that alone seems to reproduce this egg retention phenotype. And all of these egg retention phenotypes are quantified here on the right. So we wanted to see now, we know that CNMA is downstream of ATF4, now to find out whether CNMA is actually an effector of ATF4 in the context of egg-laying behavior, ovulation. We went back to our same PERK and GCN2 knockdowns, where we see egg retention exhibited when we deplete these factors in the fat. And now if we just overexpressed CNMA, again, just specifically in fat tissue, we see that we're able to block this egg retention phenotype in both genetic backgrounds. So really suggesting that CNMA is regulated by ATF4 to promote ovulation. And so this is sort of where the project is now, where we're trying to identify, see if we can identify CNMA responsive neurons or neuronal subsets that are responsible for promoting egg-laying behavior downstream of ATF4 signaling. And so again, using GAL4 UAS-based experiments, we are probing a number of subsets of neurons that have been shown to regulate egg-laying behavior in Drosophila. And so, for example, there's a recently identified small population of neurons called the OVDNs that are reside in the central nervous system. And there's also a number of overlapping sets of neurons that are in both the central and peripheral nervous system that we're also going to assay for responsiveness to CNMA via a CNMA receptor, which is a characterized receptor in the Drosophila genome. And so this work, current and ongoing work, is being done in collaboration both at the University of Pittsburgh and at Duke University, where Kafshiraza's lab has actually recently developed a circuit editing tool that I plan to use now and in the future to further investigate the neuronal control of reproductive behaviors. So to summarize what I have shown you, we found that the integrated stress response is acting in fat tissue as something of a systemic regulator of reproduction. And we've identified at least two arms of this regulation. The first is by promoting the synthesis and trafficking of yolk lipoprotein particles to shuttle into maturing oocytes and thus promoting oogenesis, as well as the production of neuropeptides like CNMA that promote neuronal activation of ovulation regulating circuits. And so with that, I would like to thank both of the labs that I've been able to work closely with, both Deepika Vasudevan, who's at Pitt, and Kafshiraza, who's at Duke. I'm grateful again to the SDB for giving me this opportunity to speak and share my work, as well as a number of other organizations that have supported my career development. And I would be happy to take questions.
Thank you, Dr. Grmai, for the wonderful talk. It's very great, and we are learning a lot. Let's see. Yeah, we have a question from Rafael. Lydia, beautiful talk. Two questions. First, do you think then that for the ovarial arrest phenotype, not egg laying, something else than GCN2 and PERK is controlling ATF4? And second, is ATF4 also expressed in the intestine?
Okay, great. Thank you, first of all, Rafa. I'm not positive. So at this point, in terms of our understanding of integrated stress response, as far as we know, both GCN2 and PERK are the only kinases known to activate ATF4. I haven't performed the experiments in the context of the oogenesis arrest phenotype. I haven't done the experiment where I knock down both GCN2 and PERK to see if it's the same degree of arrest as just ATF4. So that's certainly something we can do. I will say that GCN2 and PERK seem to affect egg laying in concert to the same degree as depleting ATF4. So my hypothesis at this point would be that those are probably the two inputs. Is ATF4 expressed in the intestine? It has been, ATF4 activity has been seen in intestinal stem cells, and it is shown to be responsible for regulating intestinal stem cell homeostasis. So.
Thank you. We have a second question from the Luth. Interesting work and beautiful presentation. Do you find the ATF4 binding in proximity to the PERK and GCN2 in the ChIP-seq data? And have you performed the RNA-seq on ATF4 mutants?
Yeah, so we haven't looked, or at least I haven't looked at ATF4 occupancy on the kinase loci. So I don't have an answer to that question. To my knowledge, it does not regulate the kinases, but I could be wrong about that. And we are in the process of looking at downstream regulation from ATF4 with next-gen sequencing. So we haven't done those experiments yet, but yeah, some ongoing work is definitely going to see what else we can identify downstream of ATF4. So.
Thank you. We have next question from Michelle. Great work. I was wondering if the defects in the ATF4 mutant interact with the ecdysone steroid hormone signaling pathway to arrest oogenesis.
That is a question that we also had. The answer is, in the context of oogenesis arrest, the situation seems maybe a little more complicated than I've been able to dissect so far. What I can say is that ATF4 has actually been pulled down in a co-IP with the ecdysone receptor with one isoform of ECR. So it does seem to have the potential to cooperate with ecdysone signaling, but we're sort of in the process of figuring out all of the details of that.
All right, thank you. I have a question from the Alexandra. Wonderful talk, Lydia. So interesting. I may have missed this, but why do you think that ATF4 RNAi does not, as far as I could tell, they also lead to the egg retention phenotype?
Yeah, good question. Yeah, so we do see egg retention with PERK and GCN or GCN2 knockdown, but with ATF4, you are correct that the number of eggs per ovary was similar to controls. We think that that's because ATF4 is downstream of both PERK and GCN2, and we think that depletion of either kinase is likely a partial loss of ATF4, where the knockdown of ATF4 itself might be stronger. And now the oogenesis rate has decreased so much that it's sort of can't show, we can't actually see the egg retention. So we have done PERK GCN2 double knockdown, and we don't see egg retention anymore. So we do think that that's just a product of partial loss of ATF4, just enough to see the egg retention, but not enough to kind of obliterate oogenesis rate. That makes sense.
All right, thank you. We have two more questions from Sandeep. Beautiful talk. Is there any expression of the ATF4 in brain? If yes, does the region somehow relate to the reproduction?
So I think I'm not sure the answer to your first question, if ATF4, how active ATF4 might be in the brain. I have not looked myself. Many of the experiments that I showed today were with fat-specific knockdown, so it might also have a role in the brain in regulating reproductive behaviors, but I think we haven't assayed that in the experiments that I've done so far.
Okay, thank you. I have one more question from Lily. Does ATF4 only affect lipid transport from adipose tissue to the follicles?
I'm not sure if your only means that it's only affecting lipid transport and nothing else in the adipose tissue. If that's the question, my answer is probably not, but I think we're still in the process of figuring out what is ATF4 regulated in the fat and what other processes it might be regulating.
Okay, thank you. We have one more question from Sharma. Do you have how many of the retained eggs remain viable? And if they are able to develop into embryos? I have the same question actually.
Yeah, it's a good question. We haven't tested it carefully yet. Just from looking at the collection vials as I'm doing the dissection, I can definitely say that some of the eggs do hatch. Some of the eggs are laid and they hatch, but we haven't looked carefully to see how many of them, what percentage of them hatch, and even like how healthy the offspring might be, or if they can become adults. So to be determined.
Okay, thank you. If anyone has any further questions to the Dr. Grmai, please feel free to type questions on the Q&A box. Then Dr. Grmai will continue to answer your questions through the second talk. I actually dismissed a little bit of questions because they are kind of overlapping with previous questions.
All right.
Thank you so much for Dr. Grmai.
Okay, great. Thank you for that great talk, Lydia. That was really nice. So it's my pleasure to introduce our second speaker today, who is Dr. Donn Van Deren from Dr. Mario Kopecky's lab at the University of Utah. Donn received his PhD also from the University of Utah in the lab of Dr. Mario Kopecky, and his dissertation was titled "The Developmental Origin of Functional Heterogeneity of HOXB8 Microglia." During his career, Donn has won several awards, including a Sciences Art Award from the 3i Annual Symposium Art Contest at Utah. For his postdoc, Donn is continuing his work on microglia, as well as some work on hematopoiesis in the Kopecky lab. And today he's going to tell us about his project titled "Investigating the Connection Between Ontogeny and Functional Heterogeneity of HOXB8 Lineage Microglia." And with that, it's my pleasure to introduce Dr. Donn Van Deren.
Okay, I am on now. Well, first of all, I just want to say Good Friday to everyone, and thank you, Leslie, for the warm introduction, as well as the organizers for inviting me to share my research on this very fruitful platform. I also want to say Happy Veterans Day to everyone who is a vet. So today, I'm going to share with you a provocative story about a subpopulation of microglia called HOXB8 microglia that not only have a distinct ontogeny, but are biologically relevant with behavioral consequences. And so by the end of my talk, I hope everyone will be able to gain a better appreciation of microglia, their function, as well as their development in healthy and diseased brains. So what are microglia? The microglia are the resident innate immune cells of the brain, and each cell contains a cell soma with ramified processes projecting from it. Now, what's fascinating about these cells is that their processes are dynamic in nature and are constantly surveying their local environment. For example, shown here are fluorescently labeled microglia in the brain of an awake adult mouse, with ramified processes dynamically extending and retracting, making transient physical contacts with neurocommunication junctions called synapses. Now, these cells are found throughout the brain, and they're distributed in a uniform fashion called cell tiling, meaning their somata, which are encircled in red here, don't overlap with each other in order to maintain efficient surveillance of the brain. And because microglia are constantly surveying the brain, making contact with synapses, what could these cells possibly be doing? Microglia have a plethora of functions involved in brain development and homeostasis, which include, but by all means are not limited to, programmed cell death, the clearance of dead or dying newborn neurons, as well as the pruning of developing axons and synapses. Microglia also have diverse physiological roles, such as regulating neuronal and synaptic plasticities. However, when microglia activity becomes aberrant, they've been associated with neurological disorders such as Alzheimer's and Parkinson's disease. It can also cause depression and anxiety disorders under chronic stress. In addition, current studies are revealing that microglia have different functions in different brain regions, which suggests functional diversity. So, given the important roles microglia have in the brain, where do these cells come from? It's well known that microglia, which again are immune cells, originate during a process called hematopoiesis, which means the generation of blood cells during embryonic development. Now, let me briefly explain our current understanding of how this process works. In mice, which is the mouse model, which is the model of choice that we use in our lab, the gestational period during which the embryo develops is about 3 weeks long. And during this time, hematopoiesis begins on an extraembryonic tissue called the yolk sac. Now, this tissue so far contains 2 successive hematopoietic waves, The first wave occurs at embryonic day 7.5 and generates erythroid-myeloid progenitor cells, which the acronym would be EMPs, that are bipotent and fated towards the erythroid and myeloid lineages. The second wave occurs a day later at E8.5 and generates a second set of EMPs, where the nature and fate of these progenitors have remained in contention. Then at E10.5, a third wave begins from within the embryo in the region called the AGM and generates pre-hematopoietic stem cells that ultimately mature into functional fetal hematopoietic stem cells once they arrive in the fetal liver. Now, for the remainder of my talk, although I have data describing the Hoxbate lineage in the yolk sac as well as the AGM, I'm only going to focus on the fetal liver because progenitors from both sources are known to seed and proliferate in this tissue. So as presented, we have three hematopoietic waves, but which wave gives rise to microglia? It's widely accepted that all microglial progenitors are born in the yolk sac at E7.5 during the first wave of hematopoiesis, and then directly colonize the brain at E9.5 through the developing vasculature. Now, this quickly generated dogma in the field, but it remained unclear whether this population could account for all resident microglia and whether microglia have more than one origin ontogeny. Recent RNA sequencing studies have exposed microglia as a diverse cell population in function, distribution, as well as response to injury and disease, which may extend to their lineage origins and development. And so understanding how cell populations develop is quite important. In fact, the fetal liver has been shown to contribute to the microglial population under certain conditions, which raises the possibility that the ontogeny of microglia may serve as a potential deterministic factor for microglial function. So, does population diversity exist amongst microglia? And if that holds true, would they have a different ontogeny? Now, during my graduate studies, we generated a Hoxbate TD tomato lineage reporter mouse model to distinguish an image between two populations of microglia and their progeny. And so in these triple transgenic mice, all parenchymal microglia express the fractalkine receptor CX3CR1 and are marked by GFP due to CX3CR1 GFP expression. Now, for the newly identified population, Hoxbate microglia, CRE is expressed in all cells that express HOXB8, leading to the constitutive expression of TD tomato fluorescent protein. This marker is maintained in the cells that express HOXB8 and their progeny, irrespective of continued HOXB8 expression. And as a result, HOXB8 microglia express GFP and recombined TD tomato, which are shown in yellow, and account for roughly 25% of all microglia, whereas non-Hoxbate microglia only express GFP, which are shown in green here, and account for about 75% of the total. And so with using this lineage reporter, I embarked on delineating the ontogeny of Hoxbate microglia and surprisingly found that this population begins colonizing the brain at E12.5. Now, at the time, this result was really odd to us, and so we wondered what could account for this 3-day difference. Initially, we thought that perhaps a subset of non-Hoxbate microglia turn on the HOXB8 gene at this time. And so we tested this hypothesis using quantitative RT-PCR and found that no HOXB8 RNA transcripts were ever detected in the developing brain from E9 all the way out to E18, even extending to adult brains. It turned out that this 3-day difference was accounted for by an alternative ontogeny. So as shown in the schematic, HOXB8 microglia, like non-HOXB8 microglia, first originate in the yolk sac, but during the second wave of hematopoiesis, and then transit through the fetal liver before colonizing the brain at E12.5. Now, for the sake of time, let me describe two key experiments that supports this model. As I mentioned before, progenitor cells arising from the yolk sac and AGM are known to seed the fetal liver. And once in the fetal liver, these progenitors can be subdivided into 4 major hematopoietic progenitor populations, which include the short-term and long-term hematopoietic stem cells, multipotent progenitors, as well as the erythroid myeloid progenitor subsets. And this graph shows the percentage of HOXB8, which are denoted by the blue bars, as well as non-HOXB8 lineage cells denoted by the gray bars within each of these populations. And as you can see, the majority of each population are derived from the HOXB8 lineage as early as E12.5. So armed with this knowledge, I wondered whether these cells collectively are competent to become HOXB8 microglia. And so I approached this question by performing intracerebral transplants in neonates, where I isolated and purified HOXB8 TD tomato labeled progenitor cells from fetal liver tissue of E12 donor embryos. Now, these progenitors were then directly transplanted bilaterally into the frontal hemispheres of recipient CSF1R heterozygous and null mice. Now, CSF1R is a colony stimulating factor 1 receptor critical for microglial survival. So what that means is a homozygous deletion of CSF1R would result in the elimination of resident microglia. In contrast, a heterozygous deletion or no deletion at all would result in the preservation of this population. And so following transplants, I examined the brains of 17-day-old recipient mice to see if HOXB8 progenitors would have the ability to engraft and differentiate. And not surprisingly, postnatal brains of recipient CSF1R heterozygous mice showed very few but lucky transplanted donor cells expressing TD tomato and GFP signals that took up residence as defined by their ramified morphology, as well as the expression of a microglial specific marker, TMEM119. In CSF1R null mice, where resident microglia are ablated, I saw many engrafted HOXB8 cells, but they appeared a little funky with fewer ramified projections, which may indicate that these cells haven't fully matured yet. Overall, these cell engraftment experiments nicely demonstrated that HOXB8 progenitors from fetal liver are capable and sufficient to give rise to HOXB8 microglia in vivo. And as mentioned earlier, the bulk of hematopoietic progenitors in the fetal liver are part of the HOXB8 lineage. And so to demonstrate whether fetal liver hematopoiesis is necessary for the development of HOXB8 progenitors and microglia, I needed a way to eliminate fetal liver hematopoiesis. And so I incorporated a loss of function of MIB in our HOXB8 lineage reporter that again marks both microglial populations. MIB is a transcription factor important for establishing fetal liver hematopoiesis. And so in effect, a loss of function of MIB results in the depletion of hematopoietic progenitors in this tissue, the fetal liver, and is also embryonically lethal by E16. It's also been used by other laboratories to show that microglia are not significantly affected in the brain at E14, although there appears to be a downward trend of these cells in MIB knockouts. So in our lineage tracing system, we can use this result as an internal control to compare both microglial populations. So from flow cytometry analysis looking at progenitor populations during fetal liver hematopoiesis from E11 all the way out to E14, virtually all KIT-positive hematopoietic progenitors, irrespective of their lineage, are ablated in the absence of MIB, which shows that the propagation of these cells requires MIB function. And it's also consistent with the literature. Now, when the brains of E14 embryos were examined by confocal microscopy, something interesting popped out. The density of HOXB8 microglia was significantly reduced in MIB knockouts, at least 50% reduction. In contrast, the density of non-HOXB8 microglia were not affected, which again represent the early microglial cells that don't transit through the fetal liver as described by other labs. Now, collectively, these results propose an argument that fetal liver hematopoiesis is necessary and sufficient for the development of HOXB8 microglia. Now, a few questions I've gotten asked frequently about these results is, so what? What does it mean to have a different ontogeny? And what makes this population so unique? To answer all these questions at once, one possibility is that intraembryonic hematopoietic tissues, in this case the fetal liver, could provide HOXB8 microglia with opportunities to acquire population-specific functional diversity relative to their counterparts. So, is there a population-specific function of Hoxbate microglia, or is this second population just a red herring? Now, this question, amongst others, is my current focus during my postdoc training. So it turns out that a loss of function of HOXB8 in adult mice results in overgrowing behavior similar to that seen in humans called trichotillomania, which is an obsessive compulsive disorder. Now, work done by a talented former postdoc, Dmitry Trankner, showed that HOXB8 knockout females have a higher propensity to overgroom than HOXB8 knockout males, which can be seen in this heat map showing the extent of hair loss due to overgroom. And behavioral analysis of their time spent grooming over a 24-hour period shows that HOXB8 knockout mice, regardless of sex, spend at least twice as long grooming themselves compared to the wild-type littermates. He also showed that HOXB8 knockout mice not only exhibit overgrooming behavior, but also elevated levels of anxiety, especially in female mice. Now, because the only cells in the brain that label with the HOXB8 iris secretory driver are microglia, and the fact that microglia are derived from the immune system, an earlier study done by another talented former graduate student, Kwon Chen, showed that bone marrow transplants of wild-type bone marrow cells into lethally irradiated HOXB8 knockout mice was sufficient to rescue their behavior, and that their time spent grooming was now equivalent to wild-type controls. And furthermore, these mice had a complete restoration of fur on their chest. I mean, that surely must have been a eureka moment. So overall, these experiments nicely show that a loss of HOXB8 function is necessary to acquire these behavioral phenotypes, and suggested that there may be a direct link between mutant HOXB8 microglia function and the observed behavioral phenotypes. So how can we experimentally test this for causality? One way to show a direct causal link would be to put mutant microglia into a wild-type brain and see if this is sufficient to get the overgrooming phenotype. And so in order to do this, I had to modify the transplant strategy. My initial transplants used CSF1R germline deletion mice as recipients. And while those transplants were successful, these mice couldn't be used as recipients to examine long-term engraftment efficiency, because these mice die by 3 weeks of age due to several developmental issues. So this prompted me to ask whether this transplant technique could be used to examine long-term engraftment of donor cells from wild-type and mutant HOXB8 backgrounds in mice that have a conditional loss of function of CSF1R in CX3CR1 positive microglia expressing cells. And so following transplants, adult mice between 3 to 5 months of age were assessed behaviorally, followed by brain harvesting to examine the engraftment efficiency as well as the identity of donor cells. For microglia identity, these high magnification confocal images in the somatosensory cortex show that most TD tomato transplanted cells, which are marked in red, isolated from wild type and mutant backgrounds, have an immunophenotypic signature of microglia that are double positive for IBA1 and TMEM, both of which are microglial specific markers. This result also holds true in other brain regions, such as the caudate putamen, as well as the piriform and cingulate cortices. Now, if we zoom out and look at engraftment efficiency, something very surprising was revealed here. Mutant donor cells were unable to distribute throughout the brain when compared to wild-type transplanted brains. And this distribution defect can be seen more clearly from their respective silhouettes of transplanted microglia, which are marked by the red dots. The quantification of these sections reveals that, that whereas the engraftment efficiency of wild-type donors was about 93%, mutant donors were not as robust, with only 69% efficiency. And another surprise was that in these transplanted mutant brains, we could evidently see the interface between transplanted mutant microglia, which are shown in yellow, and resident microglia shown in green that repopulated those spaces. But here's a big question. Having a brain populated with only Hoxbate microglia is not physiologically equivalent to a normal brain with an approximate 25 to 75 ratio of Hoxbate to non-Hoxbate microglia. So I wondered if these recipient mice that now have a wild type or mutant Hoxbate microglial brain exhibited any associated behavioral consequences, particularly over-grooming and anxiety-like behaviors. It turned out that when female recipient mice were transplanted with mutant Hoxbate microglia, they exhibited the typical over-grooming of fur on their chest reminiscent of Hoxbate knockout mice. In contrast, female recipient mice that were transplanted with wild-type Hoxbate microglia or a saline injection don't. And behavioral analysis on their time spent grooming showed that mutant transplanted female recipient mice spent more time grooming themselves compared to age-matched controls. Likewise, when these mice were assessed for their level of anxiety, mutant transplanted female mice also spent less time in the open compared to controls, which suggests elevated anxiety levels.
1 minute.
Collectively, the results from these behavioral experiments demonstrate that while transplanted wild-type recipient mice showed no signs of brain damage that affects their behavior as a result of the transplant procedure, transplanted mutant recipient mice do show behavioral pathology similar to that seen in Hoxbate knockout mice, and at least up to this point, effectively establishes a causal link between mutant Hoxbate microglial function coupled with over-grooming anxiety-like behaviors. So in summary, I want to leave you with 3 take-home messages. The first is that I've provided an argument that there are at least 2 ontogenies microglial precursors take to colonize the brain. The second is that there are at least 2 microglial populations in which one, Hoxbate microglia, is biologically relevant and causative for over-grooming and anxiety-like behavior when Hoxbate function is disrupted. And so with the establishment of this robust transplant system, I'm now positioned to interrogate the functional role that each microglial population from the wild type and mutant Hoxbate backgrounds have on the observed behavioral pathogenesis. And the final message I want to leave everyone with is that microglia therapy to treat genetic disorders in the brain is a brand new experimental avenue, an avenue that has propelled us to understand the role and behavior of Hoxbate microglia in healthy and diseased brains that may ultimately shed light on the mechanisms that drive such neuropsychiatric disorders. And so with that, I cheerfully want to thank the past and current members of the Kopecky Lab for their guidance, support, and just fantastic molecular biology hands for helping me during my graduate studies, as well as into my postdoc training. I am fortunate to be in a lab where I've been able to get a broad but very well-rounded scientific training that encompasses both ends of the biology spectrum, from gene function all the way out to a behavioral output, specifically studying macrophage development to assessing behavioral function as a product of microglial perturbation. So I also express gratitude for those tuning in and listening to my talk, and I'd be happy to take any questions now. Thank you.
Great. Thanks so much for that talk, Don. I really learned a lot about microglia. So there's a few questions rolling in. So we'll start with the first. This is from Sandeep Gupta.
Yes.
Beautiful data. Wondering if there are microglia populations that express different Hox genes, and can the expression of different Hox genes determine where in the CNS they will migrate?
Yeah, that's a great question. And my feeling from, at least from what I know about HOXB8, is that may be an unexplored territory. I am not sure if there are other Hox genes expressed in the brain. But what I should mention about HOXB8 is that we're looking at lineage-derived microglia. So what I did not show you is quantitative RT-PCR data from my 2018 paper in Development that shows basically a map of HOXB8 expression during different stages of hematopoiesis. And what I show is that in the yolk sac, there is very high but transient expression level of HOXB8, but then it drastically reduces once yolk sac hematopoietic activity goes down. In the AGM, there's moderate levels of expression, and then in the fetal liver, there's negligible levels. So it looks like as hematopoiesis progresses in this layered fashion, HOXB8 expression levels go down. And then in the brain, even from deep sequencing studies that we've done, there's just no HOXB8 transcript at all. So these are purely lineage derived where they do not, those microglial cells do not express HOXB8.
Okay, great. So we have another question from Helene Cousin. Do you know if these HOXB8 positive precursors give rise to other derivatives than microglia, like hepatic Kupffer cells, for example?
Kupffer cells, I would assume so. So, yeah, another thing that I had to leave out, right, is what other cell types does the HOXB8 lineage mark? There's a recent paper that I just published also in Development back in April, where I answered that question. It turns out that the HOXB8 lineage is marking cells that initiate in the yolk sac at E8.5, as well as cells that are derived from the AGM region. And as development continues, this lineage marker that I have, it's pretty much marking all immune cells. Every single innate immune cell that I've looked at, from monocytes and patrolling and inflammatory monocytes, they're all HOXB8 lineage derived. There is a significant amount of peripheral brain macrophages that are also HOXB8 lineage derived. So this lineage reporter definitely is able to mark all cells that are arising from the definitive hematopoietic lineage.
Okay, great. I actually had a question myself. So I'm interested in brain injury and the response to the brain after stroke and injury. And so is it known if these HOXB8 microglia have a functional role that's different from non-HOXB8 microglia after brain injury or stroke?
Yeah, so a former graduate student in our lab, Shrutakirti D, she now works, I forget where she works, over in Boston. So she did some acute and chronic injury studies and compared the two populations in the response to injury. And if I could recollect correctly, when she did under a chronic injury model, I believe it was HOXB8 wild type microglia that responded to the injury in terms of cell density around the site of injury, much more significantly than non-HOXB8 microglia. But during an acute injury using a laser ablation in the brain, both microglial populations were able to reach that site of injury at the same time.
Okay, great. Well, it looks like we're right at time. So I want to say thank you so much to both Lydia and Donn for your excellent talks. This seminar has been recorded and will be available on the SDB website starting next week. So please join us for next month's seminar on Friday, December 9th, when Dr. Sandeep Gupta from UCLA and Dr. Harini Iyer from Stanford will present. Thank you all for attending.